one of the question is asking whether or not, because we have found that REST expression is reduced in astrocyte, in neurons, in many different cells derived from Down syndrome. So the question is, what if I give them the REST expressions back? So what if I restore the REST expression? Will these cells all behave like the normal cell? We look at the baby mice, and then we look into the brain and check, is neurons getting more? And then the glia is now reduced. The answer is yes. It shows that giving this drug can restore the imbalance in Down syndrome brain.
Thank you for joining Once Changing the World. My name is Eddie Avil, and today I'm delighted and honored to have with me Dr. Michael KH Ling, who is interested in deciphering the neurodevelopmental and genetic basis of neurological disorders. With a Master's of Science in Genetics from University Putra, Malaysia, he pursued his PhD in neurogenetics at Professor Hamish Scott's lab, University of Adelaide, followed by a research fellowship in genetics with Professor Bruce Yankner at Harvard Medical School. He's been working on iPSC models for Alzheimer's and neuropsychiatric disorders. So, Doctor, really appreciate you taking time being part of the podcast. The conversation largely is going to be about Down syndrome. So, really excited to learn about Down syndrome and what are the cutting edge tools and possibly maybe reversing this disease. It would be great if you could, maybe start with explaining developmental neurogenetics for us and how crucial are genetics in understanding how the human brain develops and functions.
Thank you so much, Eddie, for having me in your channel. And let's start with the first questions. Maybe I can explain what exactly is developmental neurogenetics, right? So in this particular field, we basically specialize in understanding how the brains develop, and especially, and we look into specifically the contributions of the genetics informations towards the development of the central nervous system, the brain itself. So if you look into the second question is of your question is how important genetics in playing a role in understanding the brain and perhaps in the disease and eventually in treating them. I guess it plays a huge role. The reason is that the neural tubes, which is the foundations of our nervous system, develops in the very first month of our life after conception. Okay, from the mid first week to perhaps the end of the third week, our neural tubes will be fully formed, and these basically serve as a foundations to the development of the entire nervous system throughout the 9 months of gestational period. So at the very early stage of life, what are they to actually govern or to help the brains to develop? It will be all the genetics that encoded within our genome. Okay, and of course there are external factors like the nutritions that intake of the mothers and what kind of toxins the mothers are actually exposed to may actually change the outcome of their development and therefore predispose different people to different type of diseases at the later part of their life. And therefore, at this very early stage of development, the genetics play a very important role, and especially we call it the programmed genetics mechanism actually play an extremely important role to tell when and what cell what you should be turning into, where exactly you should moving on as a specialized cells throughout the development. So in other words, I have to say, and I personally think that genetics plays an extremely big role in predisposing someone to all kind of neurological diseases. Of course, we have to exclude those disorder that are caused by external factor like viral infections, heavy metal poisoning, and so on and so forth. So those are perhaps induced externally through the environment. But then otherwise, the genetics play an extremely important role in almost not just the brain development, but the disease development as well as almost all kind of disease that related to the brain.
Right, Doc. Thank you for explaining that. Now our brain, it's like AI has been called a black box, and brain also is pretty much like a black box, because we don't completely understand the brain, and because some 80 billion neurons, 100 trillion synapses, the way they fire and wire, it brings about all our senses: sight, sound, touch, smell, and taste. And then obviously, we thought that it's basically the neurons, the synapses, the brain's doing thing, and now you're saying that the genes and so if our body is so complex, so obviously, I mean, there's the proteins, the genes, and it's not just it's not just the genes. What you said, the environment, which is the epigenomes. Because for the longest time, I think the doctors thought that it's the genes, the GNA, the DNA, and the RNA, which was responsible. And then we thought that, oh no, there's another complex thing called the environment, the epigenomes. It plays such a big role, and you rightfully pointed out. I mean, the environment, the nutrition, it plays a big role. Wanted to get your thoughts on the neurological disorder. I mean, because that seems to be like a huge problem. I was going through this Lancelot neurology report, which was published in the year 2021. It said that almost like 1 in 3 people globally suffer from a neurological disease. And at that point in time, the report said it's close to 3 billion people worldwide living with neurological disorders. And largely, I think, there are so many factors, because I think the world has completely changed. And I think, the people all around the world are racing, or in a race seems to be like racing to nowhere, because there's a couple of factors going on over there. One is the digital overload today, because there is information everywhere. Just like maybe 15 years back, before the internet, there was no, the knowledge was not distributed, but because of the internet, which is a great thing. But now it seems like almost like a digital overloads of sorts. Then there's the social media, which is like a huge problem. The social media manipulates you, and it's like a digital drugs of all sorts. The education system also, which is like a pressure cooker of sorts. It's kind of pushing all the children. Then the ad industry, which is kind of selling you things, which you don't need. So there are so many factors which is involved for this growing number of neurological disorders, these overconsumption, and we're not understanding the need versus greed, because the world is, just wanting, what somebody else has, so maybe will be great if you could maybe share your thoughts on neurological disorders. And are these neurological disorders, like you mentioned, are they purely genetics, or it's also like you mentioned, the interplay of the genes and the environment?
I would like to maybe comment about the prevalence of of neurological disorder. It depends on whether it also encompassing the neuropsychiatric disorder. I know the psychiatrists and the neurologists may see the disease differently, but from the neuroscience point of view, they are the disease of the brain, right? And some people think that neurological disorder are the disease of the brain, but then these neuropsychiatric disorder is a disease of the mind. But for me, from the scientific point of view, they are actually all the disease of the brain. So when you think about that, and almost all of us will actually go through some level of stress, anxiety in our life, and therefore, I guess the prevalence becomes greater due to a couple of reasons. The disease has been there all the time, and I do agree that the changing of the current world with all the wars that is ongoing, with all the informations overloading, dopamine addictions to the informations that we have in our gadgets, all will certainly lead to certain level of neuropsychiatric disorder. But having said that, I think the acceptance and the ability to diagnose and classify the disease plays a very important role. And on top of that, the acceptance of getting diagnosed, in other words, the stigmatization of the diseases becomes less. I think people worldwide are more generally more acceptant being diagnosed with neuropsychiatric or neurological disorder, and they're openly happy to talk about it. So I guess that basically all contribute to the like sudden bloom, of this so-called disease being diagnosed worldwide. That's for one. And secondly, the questions about whether or not this disease are purely genetics. So I just have to take a couple of data from the research I have been working with. For example, in bipolar disorder, of course, the manifestation of the disease very much more happening towards the teenage years, or perhaps in their very early adulthood, right? Just like schizophrenia and bipolar. But the disease, the basis of the disease started all way before birth, that during in the womb. So there are many so-called risk factors has been identified, including genetics risk factor as well as environmental risk factor that has been identified that may actually predispose a baby or a developing fetus in getting schizophrenia or bipolar in their teenage or perhaps early adulthood. So things like drug abuse, viral infections, people who basically going on life event in their early stage of life, for example, abuse, poor family conditions that are like those children in the warfare zone. I mean, those basically predispose their brain as well as this individual in developing schizophrenia or bipolar in some time of some part of their life. So these certainly, I mean, environmental factors has certainly plays a role. But then you ask the questions about why are some people also exposed to the same risk, but they do not actually develop the disease when you compare them with the gender or age match group. So perhaps, if they have gone through the same processes, why are they not getting the disease? And that is basically very much more determined in the genetics, right? So there are two resistant here. One is genetic resistant, one is the environmental resistant we are talking about. So although we are exposed to the environmental, to all the risk factors, the viral infections, to all the drugs, to alcohol, to cigarettes, but at a very early stage of development, but due to a very good genetics resistance, they may actually reduce the risk of the fetus or the baby in getting the diseases in the later part of their life as compared to those basically have a different genetic makeup where they cannot actually resist uh, most of these stress that are given to them in a very early stage of their life. So this is something I have to say. When back to the questions about whether or not neurological disorders are purely genetics, the answer, if you ask me, I can be very biased because I'm from the genetic basis. So I'll say, yeah, everything is actually due to the genetics, including the epigenetics alterations to our genomes, right? That is also in response to the environment. So but then, not everyone respond in the same way. And the question is the same trigger, but everyone respond differently. Why do they respond differently? Because it was all encoded in the genetics. I think that was something I I take as a yes. They are, genetics, majorly genetic, but they are not solely genetic. So I'll have to say genetics play a certainly important role in that sense. But there are neurological disorders which are induced, for example, those fetus has been exposed to viral infection like Zika infections, they have cognitive impairments, they have other issues which are not genetics, but it was actually affected in their development because of the viral infection. There are those who are exposed to toxin, alcohol, fetal alcohol spectrum disorder that affecting the young ones, especially when the mother consume alcohol at the time of bearing the children, the baby. So that may actually lead to cognitive impairment. And a lot of these are basically non-genetics, but they are caused by the environmental or, intentionally instance of toxins.
So, doctor, I mean, what you're saying is it's largely the environment and also the genes. I mean, it's also the traumatic situations that they've gone into, so you're saying that there are people who are predisposed to that condition and who are resistant to that condition also, basically because of the genetics, the genes that they have. There's this neuroscientist, he's written, Robert Sapolsky. He's written this fantastic book called Determined, that everything is determined, like or genes and everything. So that's like I mean a good thought. I mean, if people who have not read that book, it's like a beautiful book, and you should kind of read that book. And now Down syndrome, it's a condition maybe many are familiar with, but perhaps not in depth. Can you like explain the underlying biological mechanism of Down syndrome, and what are the primary causes that lead to this chromosomal difference? This 21 extra chromosome over there?
So I guess in terms of Down syndrome, everyone knows what is Down syndrome. Perhaps they have come across Down syndrome individual in their lifetime, and this is one of the most common genetic cause of intellectual disability known to human history. And they are basically the cause of Down syndrome is very simple. Everyone has a 46 chromosomes in every cell. But perhaps, I mean, 23 pairs, okay, 22 pairs, which are non-sex chromosome, and one pair of sex chromosome, and everyone has 46 chromosome, and people with Down syndrome basically have that 47 chromosome, which the chromosome 21 has been triplicated. Instead of having two copy of chromosome 21, they ended up with three copies of chromosome 21. And not all Down syndrome have three copies. Some of them have partial. Some of them may actually have a mixture. Okay, so there are three forms of Down syndrome. Number one, which is commonly seen around us, are we call it full trisomy. People with full blown, three copies of chromosome 21 in every single cell in their body. So this is what we call full trisomy 21 Down syndrome, and these are basically happens in our so-called gamete cell. I mean, in our offspring cell, right? Or gamete cell either happens in the father or the mother, and all those research has shown that it is more likely happens in the mother from the maternal side. But then they are about 3 to 4% of the Down syndrome individual. Basically, do not they have a Robertsonian translocation type of Down syndrome, where the chromosome 21 fused with usually chromosome 14, and then as the cells divide, they still make the original two copies of chromosome 21 and the same two copies of chromosome 14, but unfortunately, one of the chromosome 14 already have additional chromosome 21 attached to it. And therefore, now they also have additional copies of chromosome 21, but carried by chromosome 14. So a small proportion of Down syndrome individual, and perhaps these are the one that have a milder form of intellectual disability, and these are known as the mosaicism Down syndrome or the mosaic Down syndrome individual. So in this individual, basically started to have aberration. The cells start to divide in a wrong way, and leading to additional copy of chromosome 21 after the fertilization, right? So a lot of the Robertsonian as well as the full trisomy 21 Down syndrome happens at our gamete before conception. But then the mosaicism happens after conception. And maybe as the cells divide, a small proportion of the cells basically divide and produce three copies of chromosome 21. This to the person having the individual of Down syndrome with mosaic condition. Part of it basically have normal cell with two chromosome 21, and part of their cells will have trisomy 21, three copies of chromosome 21. And this gives them a milder form of the disease instead of full-blown conditions of chromosome of Down syndrome. So that is the reasons why people is getting Down syndrome. And the causes is very easy. Too much of chromosome 21, and basically, there's a few reasons to it. So when you have too many chromosome 21, we assume that too much of genetic materials is not good. Okay? So that leads to the disease development. And there's another school of thought is that because you have too much of chromosome 21 as the gene expressed and too much protein produced from this particular chromosome, the entire genome, the rest of the chromosome, trying to compensate for when you have too much, right? It's a balance of everything in our cell in order to survive. So when you have too much of certain protein being produced, so the entire genomes react to that. So when the entire genome react to that, it also leads to perhaps disease and also manifestation of all kind of clinical symptoms. So the cause of it is always chromosome 21. Too much or like having extra copy or partial copy of chromosome 21 is the reasons why Down syndrome is actually being manifested in this individual. So that would be the answer.
Right, right. Doctor, thank you for explaining, and breaking it down, the three different types of Down syndrome, and the largest cause of it, which is the extra chromosome over there. So we largely kind of understand this disease and the cause and effect of this disease. So what we have right now, I mean, is these ways to kind of understand the disease further. There are tools, tools like, brain organoids, and then there's this TS1CJE mouse model. Will be great if you could maybe, make my listeners understand what a brain organoid is, how do you kind of make this, and then what is a TS1CJE mouse model, and how do you use these or other cutting edge approaches to advance your ability to understand, diagnose, and maybe possibly, treat the Down syndrome.
Certainly. So maybe we start with the mouse model first. Okay, there are actually many mouse model that are currently in our academic pool. TS1CJE mouse model is one of them. Is the one that I use a lot in my lab. I'm not the creator of the mouse model. The mouse model, if I'm not mistaken, it was actually created in University of California. So we have the model in our lab, basically trying to study the learning and memory consolidation in Down syndrome so-called context, because the TS1 CJE mouse model basically have only partial triplication of chromosome 16. So in the mouse, chromosome 16, it is equivalent to chromosome 21 in human. Okay, so mouse and human, they share about 85% of the protein coding gene. So we are actually very similar at the genomics level. And therefore, whatever that is translated in the mouse, it potentially can be used to explain what is happening in the human. And this particular mouse model basically has learning disability. Okay, if we actually put them in the pool of water and then put a hidden platform, so mouse naturally there's a natural swimmer. They will try to escape, right? They are afraid of water. So when we put them into a pile of water, they will try to swim around, and randomly they will come across an escape platform that we hidden under the water surface. So once they are there, they will be looking around. They will be staying on a platform. They will look around, and we put different cues, right? Different cues, like different signal, like a triangular shape, like a star shape, a moon shape, so that this shape is like our so-called spatial cue in our real life, like when we go to somewhere. So we remember there's a big tree and around the corner, there's a big billboard on the other side. So this is like a navigation cue for the mind, for the mouse that we are doing. Yeah. So as we train them between the normal mouse versus the TS1 CJE mouse, the TS1 CJE mouse or the Down syndrome mouse model, they basically not able to remember as well as the control mouse. The control mouse can remember perhaps after 5 days of training, but the Down syndrome mouse are having ability, these are having a difficulty in learning where exactly is the escape platform. So that is one of the very common tests that we are using to understand what is happening in terms of the way they learn and the way they navigate, and whether or not they can remember all the visual cue around them. So that is the mouse model, and there are so many mouse model that with different extensiveness of representation of chromosome 21, the human chromosome 21 on them. So they are TS65DN, they are TC1 mouse model. They are kind of different coding. Basically, they are all defined by how extensive they can model the Down syndrome full trisomy 21 in the mouse model itself. So in other words, TS1 CJE only able to model up to 40% of the genes within the chromosome 21 in the mouse. So they are not the best model, but it's best good enough to study learning disability. Whereas the brain organoids, right? The words organoids come from like the organs. It means like an organ-like cell that we grow in on the dish, so that we can understand what is happening. And when it comes to organoid, the starting material of growing organoid is the iPSC, or what we call induced pluripotent stem cell. And these stem cells are usually acquired from patient. So we have Down syndrome derived iPSC, and we also have control, means that the people without Down syndrome, and we also harvest their harvest their cell and then turn them into induced pluripotent stem cell. So from this stem cell, because as the name implies, it's a stem cell, right? It's like our ancestor cell. So by giving them the right signal, they will turn into whatever cell that you want. So in this case, we can grow them into a brain-like structure. So about 2 months old organoid, brain organoids that we grow on a dish, it basically it basically have a very similar so-called developmental as well as molecular signature as compared to the early second trimester fetal brain in development. So it basically can mimic what is happening in the early stage of development, and it is an extremely good model that you can use to study structural as well as cellular and molecular changes and comparison between what happened in the normal the brain organoids derived from the normal individual iPSC versus the brain organoids derived from the Down syndrome so-called derived iPSC. And these particular organoids are very powerful because in most of the time we see research are done on pure neuronal cell, a pure, glial cell, which are the supportive cell in the brain. So sometimes that is not how brains works. Brain works with a lot of different cells communicating with each other, and the brain organoids allows that to happen. So the organoids, as the name implies, is an organ-like structure. So they will have the neurons, they will have the glial cell, the supportive cell, and over the time, you'll find that these neurons starts to make connections. They start fire, impulses. And in one point, there are many people actually studying whether or not the brain organoids can in fact learn and think and also remember, although that you grow them on the dish. So that is the extensiveness of how brain organoids can be used to understand a disease like Down syndrome. So I guess that is what we are doing right now. And just to throw in a very quick so-called finding that we have is that as we grow the Down syndrome organoid versus the non-Down syndrome organoid, so the Down syndrome organoid basically grows faster at the beginning and then they slow down and become smaller. So these basically correspond very well with the anatomical anomalies that seen in Down syndrome individual. They have smaller head circumference, they have reduction in terms of volume and size in many different parts of the brain. So as we grow the brain organoids from Down syndrome, they also have a smaller group, smaller in size and area, as well as in terms of area and also volume. So in other words, okay, it's a very it's a model that you can now recapitulate what is happening in the patient where we harvest the cell from. So instead of study a commercialized, a generic cell line that derived from one person and imply the outcome to the whole population, now you can study specifically on patients who are still living. So you can take the cell and understand what is wrong with this patient by recapitulating so-called events that you can't see because we have missed them because we did not know they are Down syndrome or we do not know someone is getting Alzheimer disease, for example. We only diagnose them at the age of 70 or early 80s. But the problem is, if you want to ask the questions, what is happening? What caused that? We can't tell because we can't rewind and interview the patients and find out what have you done 40, 30, or 40 years ago. So one of the way is we can harvest the cell and let them have the chance to regrow from beginning of life, right? So and see what happened in their development, and that basically imply what might have gone wrong to them, and that will give us the answer. What exactly leads to Alzheimer disease because Down syndrome individual had early onset AD. So they are actually a very good model to understand genetic models to understand why AD happens and how genetics contribute to that because 100% of Down syndrome individual will develop AD towards the later part of their life. Not like any one of us, we might have risk depending on how well we sleep, how what kind of toxin we introduce to our brain, how well we preserve our agility of our mind and our brain cell. But then in Down syndrome individual, no matter what you do, you can delay, but eventually they will develop AD-like pathology. So that's the reasons why it is very important that we are be able to take the induced pluripotent stem cell from patient and regrow them from beginning to understand events that we have never been able to see in the patient. But now we can able to see them in on the dish on the cell that is specifically come from the patient. So if you don't believe this event in one patients, we can collect cells from 2 patients, 3 patients, 10 patients, 20 patients, and then you'll be able to see them collectively. That's the same event that I say that causing Down syndrome symptom. Okay, let's say learning disability is actually caused by event A, for example. So you will be able to find that by looking at the collective outcome of the many iPSC-derived brain organoids in the lab. So that is the powerful part of the brain organoids.
Right. How cool is this, doctor? I mean, thank you for explaining it so beautifully. The TS1C JE mouse model, and the mouse model, like you explain, 85% of the genetics of mouse to a human is similar. And that's how you studying, one of those TS1CG is one of those mouse Down syndrome model. And then you spoke about the brain organoids. Here you take you take a normal cell, you turn it into a induced pluripotent stem cell, and then culture it into a individuals that organoid, whether it's like a brain organoid, and the technology is expanded so much that you can take a cell and then culture it and grow it into a organoid. Here we're talking about like the brain organoid. So you are taking a cell and creating a brain, a mini brain, and in a petri dish you're kind of poking and tinkering around and understanding the individual better, that what can you learn from this, and what kind of drugs can you kind of put it and maybe understand what treatment could work. There's always hope that these R&D that you do on brain organoids, or these mouse models of Down syndrome, that eventually that they might possibly translate to humans. How realistic is that expectation in the case of Down syndrome therapies?
So I guess in terms of understanding the disease as well as treatment, the induced pluripotent stem cell has certainly revolutionized the way we actually understanding the disease mechanism as well as the applicability of our findings to the population. People has been arguing, mouse is still a mouse. We are human. The way the mouse react to the drug is very different. And of course, because of that, in earlier days, like, 10, 20 years back, people actually using a 2D model. We have the so-called cancer origin derived neuronal cell line that comes from one person, right? And everyone in the world they are basically using them. So these cell lines basically grow into neurons, and you can test all kind of drug, efficacy on these neurons. But the question is, then people still argue neurons are homogeneous cells and brain doesn't work that way. You only have one cell type in there. You need to study these on multiple cell type. And at the same time, people also argue this is actually derived from a cancer, a patient of cancer origin. So it is not real, neurons that you are talking about. So to balance the two, we want something which is relatively complex, but they are not as simple as a single cell type. At the same time, they are not actually mouse origin, because people don't believe mouse, because many drugs that has been tested on mouse basically do not have the same efficacy in human. So I think that is where the induced pluripotent stem cells come in, and the derivative of this iPSC, like the brain organoids or intestinal organoid, lung organoids, kidney organoids, and they basically give you the more complex cell type in there, okay, because they mimic the developmental process, they mimic the function, which now you can test on it, and these are human origin. Okay, and more importantly, they are not just human origin; they are patient origin. They are really patient having the disease. Now we are using their cell to test on particular drug. So whatever that you actually manage to so-called harness from the treatment on the efficacy testing or toxicology testing, so these findings becomes relevant to the patient or the people who suffer from the same disease, in general, because we get rid of the issues about mouse as the model, and we get rid of the issues about too simplistic model with only one cell type. Now you have multiple cell type. You have a clump of cells that mimicking the brain, and it also carries the primitive brain function. So if you test a drug on this patient-derived organoid, so if they have reaction and and react very well to it, so perhaps the drug will react very well. I mean, the patients will react very well to the drug directly, and of course, the finding becomes directly relevant to the people who basically having the disease. We no longer have to translate the finding in the mouse physiology and trying to make sense of it by compromising the differences between the mouse and human physiology. So, in fact, FDA has made it very clear mouse testing is is still relevant. But having a human testing on human cell, such as the organoids, is actually becoming much more important in order to understand a particular drug, or in fact to push the drug to the market for clinical trial. So that is actually how organoids can contribute to that. And of course, as you talk about Down syndrome, if you ask me, does brain organoids learn, and how do we test intellectual disability in this mini brain, right? So it's doable. It's doable. So we can still put in electrodes into this organoid and study the electrical profile of the organoid. Yeah, and we define learning in the brain, or in the electrical signal, as something as improve a response to a particular stimulus. So we can keep on shocking the brain and they start to learn. Okay, and as we let them rest for a bit, and then you can shock them, and the next time if they learn, they will give you a amplified, or we call it louder signal, signifying that particular learning process has actually happened. They have consolidated a way to basically react faster and better to the same stimulus that we have actually applied to them. So that will actually give us an indications that, oh, this drug does improve potentially the signaling in this mini brain, which is then translated in human as better learning. And you can then goes back to the mouse model to understand how this drug is now, whatever we have seen in the organs is now also can be recapitulated in the mouse. To a certain more advanced method, you can also grow this human organoid, because people say organoid is still, is a clump of cell outside the dish. So it's not in the system, there's not in the system. So people has been also embedding this organoid into a mouse brain. So you can drill a hole in the mouse skull, and then you can embed this organoid in the mouse brain and see how well these human cells react to in a systemic condition. So now it basically fulfill everyone's request. You have a systemic system where carried by the mouse as a surrogate, but the growing cell, the cell that we are studying, is basically the mini brain derived from the patient themselves. And now, whatever you're trying to study will certainly make a better relevance back to the patient, to human ourselves, instead of trying to translate their finding from one model to another.
That is so, doctor, so cool and so very exciting that at the cutting edge of frontier of technology and science, we are doing these things which is like pushing beyond of what's possible. And eventually, with these approaches where you mentioned, maybe creating like these brain organoids. These are mini brain organoids. Eventually, I'm assuming that with more learning we'll be able to create like maybe larger these clumps of these brain organoids, which maybe more neurons, more synapses, and then maybe possibly you said like putting it into a system where you are inserting it into a mouse brain itself. And yeah, how I mean, then it functions in a system where it's supposed to be, where it's not out in a petri dish, and eventually I'm sure we'll be able to understand this complex body and the complex brain. And once we understand that, and if we can just chip at maybe taking these diseases out, because this is so draining to humanity and mankind. I mean, all of these diseases, and these neurological conditions, and these age related diseases, and if we are able to kind of better understand the human body, the genomes, the proteins, the neurons, and the where they fire and wire, eventually if we kind of understand this, I'm sure we'll be able to like make a dent and possibly start maybe not just maybe treating a disease, but eventually maybe like finding cures, where we reverse diseases. And I'm like really really excited for that world, and kudos to researchers, scientists such as yourself. So doctor, you recently authored this paper on restoration of rest protein in the brain, which inhibits reactivity of Down syndrome in iPSC derived astrocytes. Could you unpack this for us, and what this means, and why is it such an important step?
Okay, so maybe I should start with REST. All right, REST is actually protein. It stands for RE1 silencing transcription factors. So, of course, if you look into the literature, uh, they're actually an indications of reduction of this RAS protein in Down syndrome derived cell like embryonic stem cell and also the primary neurons that indicating that this protein has been reduced. But there's actually a huge gap in between, almost like a 15 years gap where no one actually looking into this protein. And why it comes to my interest is that when I was actually doing my fellowships back in Harvard Medical School, we basically look into this RAS protein in Alzheimer disease, schizophrenia, and bipolar disorders, individual derived iPSC, which we make them into brain organoids and trying to understand what is the role of RAS. And to our surprise, if you look into the post-mortem brain of people with AD, for example, right, in Alzheimer disease, versus those post-mortem brain of centenarian, who will live up to 100 years old, but they do not develop AD. So if you look into their post-mortem brain, especially at the frontal cortex, which is the regions where control the cognitive function and higher executive function of us. So we find that the RAS expression in centenarians, those who have a very good cognitive so-called preservation, they have this RAS protein in the neurons, inside the nucleus of the neurons, okay? So they are very well preserved. The neurons are beautiful. But then in Alzheimer disease individual, if you look into their brain, the postmortem brain section, this particular protein is no longer in the neuron, but it has been sidelined. It is everywhere else except within the nucleus of the neuron. So we have this idea, and of course, like what you mentioned, understanding the disease, we need to understand the entire picture, and we do a lot of transcriptomic, the analysis of the RNA, the analysis of the DNA, and we found that this particular expression of protein, the so-called RNA, it never changed. But it was the localization of this protein change, right? So instead of in the nucleus, is not in the nucleus, in the cytoplasm. And that basically correlate and they segregate very well with the disease. People who are healthy and live up to 100 years old without, cognitive impairment, they have very good rest in the nucleus. Those with AD, they have rest inside the cytoplasm, but not in the nucleus. And because of that, uh, as my interest is always in Down syndrome, I look back and I found this paper where indicates RAS expression has been reduced. The paper was actually published by Dean Zetic from Queen's Mary University, London. So their group is one of the pioneers group, basically indicates RAS is reduced. And now I continue on their works and looking into how RAS basically potentially is the reasons why we have many type of pathology actually see in Down syndrome. So one of them is basically the astrocyte. We actually look at two paper published within this year related to RAS, and one of them is on the progenitor cell, which is related to how the cells are shifting from instead of become a neuron, they turn into glia cell. And one is the study on the astrocyte. And all these cells are derived from the human induced pluripotent stem cell that we got from Down syndrome versus the control, the normal individual. So when you compare the two of them, right? So this is very well-known biology in Down syndrome. They are not only having they have less neuron to start with, they have more astrocyte, and not only that, the astrocytes are more reactive. So when astrocytes are more reactive, it means that the brain is in constant inflammation mode. Their brain is continuously in an inflamed situation. And that leads to a lot of other issues like neuronal cell death. And eventually, they will have, they fail to replace the cell. And eventually, they will drop into accelerated aging as well as accelerated or early onset Alzheimer's disease. This is what we believe the trajectory is in terms of their brain development. So what happened is that one of the question is asking whether or not, because we have found that RAS expression is reduced in astrocyte, in neurons, in many different cells derived from Down syndrome. So the question is, what if I give them the RAS expressions back? So what if I restore the RAS expression? Will these cells all behave like the normal cell? Okay, and because if the cells behave like a normal cell, then it also means that, there's a very good chances that lithium can be given to Down syndrome individual to perhaps reduce oxidative stress, to reduce the neuronal cell death, to also reduce so-called astrogliosis, where it's the inflammations caused by astrocyte itself. And in this particular study, we did actually prove that as long as we treat the Down syndrome derived astrocyte with lithium, the astrocyte basically becomes calm. They are calmed down. They no longer becomes reactive, and that is very important. Astrocyte should be only reactive at the time of injury, so that the brain can restore themselves based on the signal that they actually secreted. But if this injury persists for too long, right, the cells eventually becomes harmful and leads to normally neuronal cell death. The neurons will start to degenerate and die. So that is one of the things that we are looking into in that particular paper. And to our surprise, when we treat the rest with lithium, we treat the cell with lithium, rest expression increase in the nucleus. That's number one observation. And when the rest is increased in the nucleus, these astrocytes basically turn from reactive mode to non-reactive mode. And that is something we want to achieve. And that is what we actually reported in that paper. And we attribute this to specifically to the rest action because we have another drug known as X5050. It's a small molecule. So when we put lithium together with this X5050 target rest specifically and then cause rest to degrade. So when we put the lithium and X5050 together, the effect of lithium becomes neutralized. So in the presence of this X5050, even though you treat the cell with lithium, nothing happens. The cells remain reactive. More importantly, we also treat a normal cell, a normal astrocyte, which is non-reactive, and we give them X5050 to degrade rest. So what if a normal cells lost the rest protein? So what happened is that the astrocytes become reactive. So we turn normal cells into a Down syndrome like cell when rest is gone. And we turn the Down syndrome cell into a normal like cell when we restore rest. That is the final conclusion of the paper.
Right, very, very interesting. So you mentioning that this brain protein called rest. I mean, when it reduces, and this astrocytes, when they are more reactive, that's the main one of the main issues. And treating them with lithium and X5050 is one of those uh, approaches which you're seeing very, very helpful for Down syndrome.
Lithium treatments restore rest protein and makes the astrocyte non-reactive. But the X5050 is the reverse, is the opposite of lithium. So we give the normal cell X5050, so the normal cell behave like a Down syndrome cell because X5050 basically remove rest from the cell. So it's of opposite action.
Okay. Can you explain neurogenic and gliogenic and how targeted inhibition of the JAK-STAT pathway might influence this neurogenic to gliogenic shift?
So maybe I should explain at the beginning why JAK-STAT. So as the cells in our body, as the sperms and the egg fertilize, into one cell, and then they divide into 2 cell, 4 cell, 8 cell stage, right? So to a certain level, as the neural tube is being formed in our first month of pregnancy, our first month of conception. So what happened is that these we call it primitive or naive cell. There's many cells that remain naive. They do not know what they are turning into, right? So all the cells in our body comes from the stem cell, from the pluripotent stem cell. So at the very early stage of development, these cells waiting for the cues, for the signals from environment, from the so-called pre-programmed genetics information. What should I turn into? Should I turn into neurons? Should I turn into astrocyte? But if you look into a normal developmental trajectory of a normal individual. So these cells will first proliferate. They have to make more of themselves first. The naive cell means that the stem cell. So this is like, I just want to give a maybe easier analogy. So we put money into our bank, right? So at the beginning, you don't want to spend them because that is our capital. So you want the capital to generate interest. So the more interest you have, the bigger the capital is going to grow. So what happens is that when it comes to the right time, we're going to spend some money. So spending money is actually differentiation process, isn't it? Because the money is not coming back because we're going to spend them, they're going to turn into something. But a smart person will spend the interest, okay, for the enjoyment or to buy whatever things that they want. So you should always keep the capital to continue to generate interest so that you can sustain, throughout your life. So exactly like the same concept in the brain. So this knife cell will first generate as many as possible of themselves. So when in the right time, due to genetic so-called pre-programming and also external factor, some of these cells will turn into neurons, or some of them will turn into astrocyte. And at this time point, at this juncture, Down syndrome individual have issues where they keep on receiving informations to tell them, turn into astrocyte, turn into astrocyte. Astrocyte is a supporting cell, but less cells being turned into neuron. The neuron is the one that is going to store all your memory, that basically, execute the so-called cognitive functions and ability. So at the end of the day, Down syndrome individuals, they are born with less neuron and more astrocyte. So that is the question we are trying to ask. Does Down syndrome individual has issue, well, especially the naive cell, having issue making decision? And if they have issue, why are they making a wrong decision? And one of the reasons why they're making the wrong decision is because of this JAK-STAT signaling pathway. Of course, this is not the only reason, but there are many other reasons. But this is one thing that we have been looking into for many years. So the JAK-STAT signaling pathway is the pathway that basically telling the so-called the pluripotent stem cell or the naive cell turn into glial cell, turn into glial cell. So when you have so much of that information, right, at the very early stage of a life, so they basically depleting the capital to buy something that is not at the right time, right? You may need right, but later part of your life, but not so early at this time point. So a lot of these cells turn into astrocyte, okay, and less cells turn into neurons. And what happens is when neurons die, when neuron dies one day, you will be running out of these pluripotent cell or naive cell to repair, to replace them. So we believe this is the reason why the Down syndrome individual eventually have accelerated aging and also develop early onset Alzheimer's disease because they are running out of the capital. So the questions are back to the JAK-STAT. JAK-STAT is one of the signaling factor turn into glial cell. So this is exactly what we are trying to do. And a few years back, I think almost near to 10 years now, we actually started using a drug, a cancer drug. This is the first FDA approved cancer drug is targeting JAK-STAT signaling because we have identified this pathway, this signaling as the reasons why the cells turning into astrocyte. So what happened was we basically we take this drug known as Ruxolitinib, which is now in the market. It's a drug that basically prescribed for patient with myelofibrosis. But the number one target is this drug only target JAK. Okay, so when they inhibit JAK, then the downstream STAT is also inhibited, and therefore you can shut down or inhibit the signaling completely. So we ask the question: so if we give the pregnant mother, I mean the mice, pregnant mice, with this drug, will they produce a smarter baby eventually, right? This is what we actually ask for. Of course, before we ask the question about smarter or not smarter, we also ask the question, do they restore the neurons to the so-called glial cell ratio imbalance? Okay, so that is what our question is. So we give the mothers with ruxolitinib at the level where it is not therapeutic. Okay, we use a very low level and then we want to give them because the ultimate aim is to make it into a supplement that high risk mother will be able to take them, especially when this is the first childbearing year and they are actually at the high risk stage like 35 and above. So perhaps they can take this drug not to prevent Down syndrome, but to basically prevent intellectual disability in the case of when they get a Down syndrome child. Okay, so that is one of the bigger idea that we have. So we first by now have actually published this paper and actually shown that if we give the pregnant mice with this drug and then we analyze at two stages. At the stage of where they give birth, after they give birth, we look at the pups, look at the baby mice, and then we look into the brain and check is neurons getting more, and then the glia is now reduced. The answer is yes. It shows that giving yeah, giving this drug can restore the imbalance in Down syndrome brain. But what we have been doing is we do this on the normal mice just to see, to prove a concept. So we have actually shown that the pups at after 1 day of birth, and their brain basically express, greater marker for neurons and lower marker for astrocyte. So we have proved that. So the next question is like, we have proven that, and then the next question is, um, I mean, does it really solve the issue? Do they have do they have a better intellectual after that? So we leave the pups, some of the pups treated with ruxolitinib during the gestational period, and these pups grow up to 2 months. So 2 months, 2 months old mice are equivalent to young adult. So after 2 months old, then we subject these mice to the same pool experiment that I mentioned just now. Do they remember the escape platform? And to our surprise, that the mouse where the mums were given ruxolitinib during the gestational period significantly able to remember all the landmark and can search and reach the escape platform much faster compared to non-treated mouse. That is really, really a nice breakthrough from our work, indicating that inhibiting one particular pathway here, JAK-STAT, can reverse the neurogenic to glyogenic shift. Okay, and at the same time, it has a really tremendous outcome towards the young adult minds, indicating that they actually have better learning and memory consolidations compared to the non-treated mice. So that is something that we're actually doing. And we now testing this on the transgenic mice, the TS1CJE that we have in the lab. And this is still ongoing before we actually decide to push it into the clinical trial. Now we're actually working on the mouse model and see whether or not targeting JAK-STAT in this mouse model basically can lead to the restoration of their the spatial learning and also memory consolidation. So this is something we are actually doing now. On top of that, we also use lithium because we have shown lithium to be really useful in targeting RAS. And RAS, in fact, sitting on top of JAK-STAT because RAS suppress JAK-STAT. And the story fall into place because when Down syndrome have less RAS, they have more JAK-STAT. When they have more JAK-STAT, they have more calling to turn into glial cell. So because of that, we also think that instead of targeting JAK-STAT, we can target RAS because ruxolitinib is a cancer drug and it's very expensive to give. But lithium is a very cheap drug. I wouldn't say cheap. It's very affordable drug because it's a very old drug that has been approved like many years ago. So if you give lithium, then it will be able to benefiting much more people than actually the rich one who can actually pay for ruxolitinib. That was our current idea. So we also look into lithium, and we also give lithium to the normal mice in our lab. And this doesn't have to be in pregnancy. This can be given even to adult mice. And in fact, given the adult mice with lithium treatment, they also becomes smarter, able to remember the escape platform better than those untreated mice. So now we have actually these two discovery that we think will be very, very useful to apply them only to solve one issue at a time. That is the intellectual disability in Down syndrome, either through the very early pregnancy or after they have been born. And we have to find out what is the right dosage to be given to the Down syndrome individual so that they will not cause toxicity, they will remain a supplement, and they will have a long-term cognitive so-called health benefit. So that is what we actually trying to do right now by targeting the neurogenic to glyogenic shift issue in Down syndrome individual.
Thank you and congratulations on the work, Doctor. And I hope that this effort of yours, this work on ruxolitinib drug, I hope that kind of translates and your clinical trials, it moves from mice to humans. Because these high risk mothers, if they can use these medicines, and you mentioned, that's really expensive, and possibly not that. Maybe lithium can do the same. Then what a world that would be. Maybe where we are able to possibly maybe try and catch that right where it's a nip it in the bud, and that will be like a great thing. Now, doctor, I mean, obviously, I mean, the big provocative question, you mentioned about so many different approaches to kind of maybe understand Down syndrome, and maybe possibly understand it better, to kind of find small approaches to maybe control, or maybe reverse. Are we, from your side, are we anywhere close to actually reversing Down syndrome, or should we think more in long term of like just management and improvement, instead of reversal? What are your thoughts on that?
I think all approaches should be go on in parallel. So the very well established special education, speech therapy, occupational therapy, perhaps the well known, so-called supplementations that has been actually shown to be having beneficial effect to the cognitive ability of Down syndrome, like the phenolate supplementations, green tea supplementation. So I think those things can go on because they are actually relatively good evidence indicating it may actually help to preserve, if it's not improving, that to preserve the cognitive ability of Down syndrome. But at the same time, we have to ask ourselves, I mean, are we happy with Down syndrome being not cured? Or are we happy that treating the symptomatic management itself is actually good enough at this juncture? So I guess this is the reasons why the Down syndrome scientists, enthusiasts, I mean, we basically wants to treat, not what we wants to cure. And in fact, from my point of view, it's still a long way to go, but there are many breakthroughs on the dish that indicating that this is doable. But delivery is another issue. Okay, and this comes to one of the study which has been recently published, right, by this Dr. Roitaro Hashizume from Mi University in Japan. So basically able to get rid of the entire chromosome 21 so that you restore from 3 chromosomes into 2 chromosomes. So this is certainly a huge breakthrough. And the similar approach has been actually done. So this is more like a genetics modifications approach. But what I've been talking about using drugs, those are pharmacological approach. And there are some others which is non medical approach, which is the therapy, as well as the supplementation approach, which has been used largely by many Down syndrome community. So when it comes to approach like genetics, removal of the entire chromosome 21, there is actually a huge breakthrough. So this has been done by a different group in Harvard as well. But the one that is reported by Dr. Hashizume in Japan are targeted approach. They can target a specific chromosome 21 and inactivate them by degrading it. Okay, so this will allow the entire cells to behave like a non-Down syndrome cell. So they're now having 2 chromosome 21 instead of 3. And in fact, Dr. Hashizume group shows that cells that where they have gotten rid of the additional chromosome 21 behave like a normal cell. So a lot of these so-called oxidative stress markers or cell death markers has now becomes reduced, and they basically improve after the removal of the chromosome 21 using the targeted approach reported by Dr. Hashizume. But the question is, how far are we going to able to remove the chromosome in a full-blown individual, right? So this is all down to the methods are there. Okay, there are way to do that, but the question is efficiency and delivery. As a full-blown human, a Down syndrome individual with a full-blown full trisomy 21, every single cell in our body has 3 copies of chromosome 21. And as the paper reported by Dr. Hasizumi, they only managed to restore up to 40, I think 30 to 40% of the cell. So the question is, how much cells we need to restore? We need to get rid of the additional chromosome 21 to have the so-called health beneficial effect to Down syndrome individual. That's question number one. And question number two, how can we deliver all these genetics tools into the body of Down syndrome individual in order to achieve something is significant? So, and potentially, would there be an off-target or like a side effect? Because testing on the so-called iPSC derived cells on the dish is again never the same with testing it in human. So there will certainly be connections between Dr. Hashizume a breakthrough right now between in the dish to human. So there will be much more work needs to be done, perhaps at the level of using the mouse model for Down syndrome and see whether or not the targeted method could be used to remove the additional chromosome in this model before we actually bring them to the human study. So if you ask me how far and how fast, I'm not quite sure because we have to also bear in mind treating a full-blown Down syndrome individual is different from treating them from the womb because in the womb, many of these organs are still developing. And if no treatment is done and during the development, many of the so-called aberrations already happen. For example, the brain, which happens to be which happens to be developed from in the very first month of after conception. So, what happens if we only treat them when they are 5 years old, 6 years old, 7 years old? Are we only able to preserve the intellectual capability, or are we going to improve their intellectual capability, or what in the expense of what adverse effect? So, those are the things that we need to really bear in mind before you know, defining an approach as the successful approach in treating and perhaps curing Down syndrome. So whatever Dr. Hazishumi done in Japan, that is a curative approach. So we are getting rid of the cause of Down syndrome, and of course that should technically restore the health and the benefits in the individual. But then, as I say, a lot of damage might have already happened during development, and can we restore that, or can we prevent it from further damaging? So that is something that is still a big questions that we have to look into.
Yes, yes, doctor. So, I mean, it's so exciting that these breakthroughs are happening, and I think there's a convergence of tech stack. This CRISPR-Cas9 gene editing technology with artificial intelligence, bioinformatics. Where we are able to kind of understand the entire data, that a human produces, and also proteomics. And I think maybe that this match of things maybe will be able to kind of find more breakthroughs. And you mentioned, I mean, even Dr. Hashizume's in this breakthrough of removing the extra copy of chromosome 21. The delivery obviously is going to be a problem because these gene therapies, right now it goes into multi-million dollars. But hopefully, I mean, like this convergence of tech stack, with AI, with gene editing and proteomics and so many others, eventually neuroscience will possibly help us into maybe picking into this thing further, understanding it in a way that maybe, like you possibly mentioned, maybe we'll be able to chip away things and possibly maybe treat, not just treat but also cure, Down syndrome. And how awesome would that be, doctor? Are there any promising ongoing clinical trials right now for Down syndrome treatment or management that you can update us with?
Um, to be honest, it depends on the focus of the clinical trial. There are clinical trials that is ongoing on Down syndrome individual, but they basically very much focus on different clinical manifestation of Down syndrome individual. So the one that has been carried out, I think there are quite a number of clinical trial at the moment focusing on the acute lymphoblastic leukemia. There are also clinical trial that is focusing on the AD in Down syndrome because that is actually an issues among them. So there is really no clinical trial on the curative measurements like what we have been talking about. Removing one of the chromosome 21, is that possible? So there is really no such clinical trial at the moment in Down syndrome individual, but there are plenty of clinical trial that basically very much focusing and zooming into very dedicated issues faced by Down syndrome individual. The AD, the clinical trial on cognitive functions, and also clinical trial on cancer that is actually at risk in the people among people with Down syndrome. So there is no one clinical trial on the curative measurements of Down syndrome at the moment.
Right. Doctor, my last question to you. Your work is also in autism, schizophrenia, and Alzheimer's disease. Will be great if you could maybe talk about that. And for the parents or children living with Down syndrome, is there any message that you would leave them for them?
So far, my work with schizophrenia and Alzheimer disease in specifics were actually done in Harvard Medical School when I was actually a fellow scientist there. When I returned, I'm now very much focused on Down syndrome, but the interest on Alzheimer disease are still there because it affects 100% of the Down syndrome individual. And the model that I've been talking about in the lab, they are also, this model from Down syndrome also has the pathology of Alzheimer disease, for example. All right. So this kind of work are basically still ongoing, and the interest is very much pinpointing on the rest protein that we have been talking about. It's just that after I've returned from the state, I very much focus on Down syndrome, and we are all looking at the same thing. How could RAS basically play such an important role in AD, in bipolar, and perhaps schizophrenia? The work with schizophrenia has not been published, but the what we have been published is the bipolar and Alzheimer disease, implicating how rest, a loss of rest in this nucleus within the neurons leads to so much pathology seen in AD, AD brain organoids itself. So what exactly? I don't have much experience with autism, but just from my knowledge, autism is certainly another much more complex condition because due to the spectrum itself, that some of them have really a severe symptom, and some of them basically have a milder symptoms and potentially able to mingle and actually mix to the mainstream societal so-called norm by going through some sort of speech and occupational therapy, and some of them are really so-called severe cases and with severe symptoms. They require almost daily care, require a daily carer to take after to take care of them all the time. So I have not really looked into rest as well as other signaling pathway related to autism because the cause of autism are uh, largely genetics, but we can't pinpoint what exactly or which part of our genomes are really causing autism. But we also understand there are so-called hypes out there, highlighting potentially some environmental risks that may actually lead to autism. So autism is something still is very rampant. The research is still going on very rampantly, and at the same time, a lot of the focus are now, just like when Down syndrome first started, right? Everyone is focusing on how to solve the symptomatic manifestation. So how do we improve their speech? How do we improve their so-called assimilations to the societal norm, and so on and so forth? Okay, so that are very much the focus of the current autism study. Although they are iPSC derived from autism, so-called autism spectrum disorder individual, where we're trying to figure out what exactly has gone wrong at the genomics level, at the genetics level, trying to pinpoint what are the messed up molecular pathway in there. So that would be something that is certainly interesting to look into. But unfortunately, my labs are not looking into autism at the moment. And whereas, what is my message to parents with children suffering from Down syndrome? It really, there are many parents wrote to me asking for, how good is the research right now and how can we use it for the children. I have to say we have to be very careful. My first advice to them is, do not trust everything that you read. Even though whatever that we have published, they, you see, that's how mainstream media has been sensationalizing the breakthrough in the lab. So the breakthrough in the lab remains in the lab until clinical trial is carried out. We should not trust any treatment just yet, and try not to self-medicate. Okay, unless you are talking about natural product which has been safely consumed by many people. Yeah, you can try that out. That's not an issue. But with again, too much of everything, it can become a poison. Okay, to anyone. So we have to be very careful in terms of that. And secondly, I guess parents who have the first Down syndrome in a child or encounters perhaps the psychological burdens that they have is huge. But one of the way, especially in countries where abortion is illegal, so they have no other choice but to give birth to the Down syndrome child, despite they has been diagnosed to have Down syndrome, or maybe it's too late for them to actually carry out the abortion because most of the time Down syndrome is diagnosed in the late second trimester, and will be too dangerous to abort them because of the maternal safety. So for that reasons, then they have no other choice but to give birth to Down syndrome individual. And I guess the best advice for them is to join the interest group, to join the parentals group who also have Down syndrome individual, and they basically are having much more experience. They will be able to support, and you feel that you are not alone in this world. And in fact, for Down syndrome, there's actually there are many very established organizations and foundations basically helping parents as well as individuals with Down syndrome in many different countries. In Malaysia, for example, we have Kiwanis, which is actually the Down syndrome foundations established to help Down syndrome individual in terms of their learning as well as their speech and as well as how they take care of themselves. They also give out a lot of tips and informations to the parents to teach them how to take care of the child and what is the best way to deal with Down syndrome baby. So my advice to them is not the end of the world, perhaps is the start of a new journey with a very special individual.
Exactly. Doctor, what a profound note to end on. We live in such a great world. And I think maybe once we understand that each one of us is so very special and everything happens for a reason. And I think the world will be such a beautiful place if in the next possibly maybe, 10 years, 15 years, where all of these tech stacks converges, with the genetics, neuroscience, with artificial intelligence, and with researchers such as yourself. I mean, Dr. Hashizume, and your approach, his genetic approach with your rest protein approach. If we can kind of marry that, and finally, if we can find a cure, what a fantastic world would that, sending you all the positive energy, and that may you, find that recipe, or that cure, or that treatment which changes the world for the better. Thank you once again, and really, really appreciate you taking time and being part of the podcast. Please keep on doing what you're doing. You're making the world a better place. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you guys. Bye bye. Thank you, doctor. Really,
Yeah,
really
thank you so
appreciate
much.
this. Yeah. Thank you.
Yeah,
Thank
it's a
you. Thank you.
pleasure, yeah, to
Uh,
join your channel. Thank you so much.